So I'm going to talk about veridical data science toward trustworthy AI. I put a few concepts already brought up by the previous speakers: stability, robustness, successibility, interpretability, and causality. But I want to emphasize more, which is a reality check, which I think in this theoretical world hasn't been mentioned too much. So there was this opening talk by Alexander about the spherical cow in a vacuum. And this is a real cow in a real environment, so I'm from both worlds, and I try to bridge the two worlds. I was hoping that ChatGPT can help me to come up with my world. So I asked ChatGPT to draw the opposite of spherical in a vacuum. It was not able to do that. I don't think it understood the opposite of spherical. So I had to go back to search on Google and got two real images, well, the first cartoon. So anyway, since this is a very new community for me, let me just do a little self-intro with this spherical and real cow. So I was a statistician as a PhD student in Berkeley, did structural machine learning and MDL theory, so connect with the SLP, but before the singularity hit MDL theory. Went to information theory, did MCMC convergence, and slowly drifted into signal processing, and went to Bell Lab for 2 years and did some review papers, and that was a closure on MDL. And then the next phase for 10, 20 years was machine learning theory, bagging, boosting, lasso, spectral clustering, and at the same time going to the real field with real cows, with remote sensing, computation, neuroscience, genomics, and fate methods. We're looking at really Drosophila fate map with gene expression data and precision medicine. For the last 10, 15 years, we have been doing climate science, interval machine learning. Oops. Oh, very sensitive. So I shouldn't move, right? Is that like I have to stay?
Yeah, I think don't even breathe.
Be dead. Never solve the problem.
I'll hold my breath.
Yeah.
Netflix. Yeah, let's just pick something. Should be HTML3.
Maybe I'll sit there. Here, maybe that'll reduce moving, right?
Yeah.
We need some stability here for sure.
It was multiple TV.
Yeah. Yes. I try not to move in my chair. It's very hard. So you can see that.
Real world cow.
Exactly. But I like to go back to the spherical cow now.
Sometimes it works better.
So you can see that I'm mostly in this real world, but returning to theory through deep learning, because I think mathematical conceptual theory, relevant theory, will be very useful. And so what's the common thread, right? I'm very broad, I'm a journalist, but I think a common thread starting from my PhD thesis was really a pursuit of simplicity and relevance to understand the impact and to bridge the gap between theory and practice. I had two advisors. It was kind of a novel thing at the time, but one theoretical, one's applied. And what does veridical mean? Nobody has told me. That means truthful. And in veridical data science, it means we seek truth. We also want to be truthful to the data science life cycle. And the paper with my former student came out 5 years ago. It's really ambitious philosophical conceptual framework for practicing data science responsibly alongside rigorous documentation. The context cannot come in unless you document. We want trustworthy data-driven results with data science very much under all most of the AI development. That's why this is very foundational. We want to critically assess the results in the context, domain, and reality. And the verification of trustworthiness has to do with uncertainty. You cannot have always like exactly deterministic result. So why I want on the past? It was really about 15 years ago, there was a lot of attention on replication crisis, especially in oncology. And the problem persists. A couple of years ago, 73 teams of social scientists worked on the same data with the same hypothesis and got very different results because they made different analytical calls. They cleaned the data differently. So they get very large positive and negative result about immigration and social policy support. And there is a similar paper in biology. And I did this exercise in my class. We asked students to clean data with the same rubrics. And they end up with different data sets. And they have different assessment about sensitivity, means the patient who really need a CT scan is in ER room. What's the sensitivity? Make a difference if sensitivity is 90% or 80%. So that means that data cleaning is a huge source of uncertainty. And data science is never just one step, right? Mostly here in the spherical world, you worry about the algorithms and the math. But in fact, if you are a patient being treated by algorithm, I hope you care about all the steps, not just about the mathematical bits. And there are many, many sources of uncertainty, a lot dealing with human judgment calls. It's like a perturbation with last is about linear perturbation in physics. Here's human perturbation. So this is a very high overview of what we have done for the last 10, 15 years. And both from new machine learning methods, I'll show you 2 case studies in biomedicine. And we have some theorem. And then we do uncertainty quantification and resources, and hopefully the theoretical minds will help me to find the theoretical foundation for veridical data science. So, as I mentioned, we really try to marry machine learning with statistics, right? My late colleague Leo Brahma had this paper called One Culture. This is tried 2 cultures. This is try to make it one culture. Put ideas from machine learning, predictability, computability together with uncertain quantification, but at the same time build a big tent, not just sample-to-sample variability, but consider other perturbation to the whole data science lifecycle. And it's a unification, a synthesis, expansion, and try to build uniform, universal language platform for further development. Very much coming from the trenches, but now I like to see more abstraction and framework. So predictability is from supervised learning and for reality check. That's machine learning took heart. And now we have expanded to unsupervised learning, so P is not really for reality check, but I'm stuck with P, so it's still called PCS. And stability is this common sense idea that you want to shake the whole system in a reasonable way, and maybe measure the stability or perturbation result in multiple ways, and then you aggregate sensibly and for achieve stability or add stability to the process. I cannot do it in a vacuum, actually. This has to be done in context. That's why documentation has to be there. It doesn't belong to the vacuum, but belongs to the practice of data science.
Maybe you'll give one later. What's an example that would be like coming from the domain?
In fact, I will get there. Yeah. One example is more just machine learning. So I fall in love with stability in this paper. Really try to put data perturbation together with model perturbation, right? And linear model, you interpret the linear coefficient, but I'll show you that the interpretation goes beyond just a linear kind of response theory. And the stability principle really try to build a very big tent, so we can unify different forms of perturbation and use the documentation to see which ones are reasonable and relevant for your particular practice. And it's really actually stability is the heart of the statistical theory, classical, right? Central limit theorem is stability result. And random matrices result, you can see that you don't have Gaussian distribution in the end, but you do perturbation in the proof. And uniform stability result in machine learning bounds the generalization error. And the principles really unify many aspects of trustworthy AI, and may also, I think, safe AI. Reliability, susceptibility is stability, robustness, stability, transfer learning is a form of stability, fairness is about subgroup stability, differential privacy is a form of stability. So all of this can be put in this big umbrella, and you can choose and pick. And I argue that PCS is practical prerequisite for practical interval machine learning and trustworthy AI and AI alignment and for causal conclusions. So one thing we haven't heard, I think for me, interpretability is relevant to a human audience. And in medicine, right, my model might be interpretable to me, but might not be interpretable to the patient or to the doctor. So that makes a difference. The relevance is what we put in, right? You have predictability for model checking. If model cannot beat random toss, I don't think you want to interpret it for any practical purpose. You might want to diagnose the problem, but not for dealing with a human audience. And descriptive accuracy is very, very vague. So you have to use the audience to know what's descriptive enough for the audience to know what's enough. So we had this paper from 2019 really put forward these three principles: predictability, descriptibility, and relevance, and has been, I think, almost 2,000 times. So this is really bring together the expertise by group from different domain areas and to put together what we're going to define as interval machine learning. So the stability principle has two roles. First, you assess whether things are stable enough for your purpose, and then you might do new machine learning algorithms, so AI algorithm, to add stability so that achieve your goal. So assessment and improvement. And people usually ask me, how do you do perturbation? Because if you do everything, you never go home. It's a combinatorial problem. So you still have to have high-level human judgment calls. And you write a document about it. You cannot remove human judgment calls. We always do human judgment, but you document it and you make an argument why these are reasonable human judgment calls. And there's something in the Bayesian literature called The Garden of Forking Paths by Gauman and Loken. They reckon they call multiverse. They recognize the same phenomenon that you have this forking, many choices, but they don't give principles to know how to work this path. So the phenomenon is recognized also in that literature, but I think they're also pushing upstream. So I started as a modeler, didn't worry about data cleaning. Now I see stability very much unifying at every stage. Even for problem formulation, we have people from physics, statistics, machine learning, math. Sometimes languages actually are not the same. I talked to a cancer researcher once over dinner, and for her, matrix meant a microenvironment in the cancer near tumor. And for me, matrix is a tabular. I gave up in the end, try to explain that I have a different definition of matrix. So you can see that linguistic stability is right at the center for multidisciplinary research. So people usually in real data, they say location, location, location. Here is documentation, documentation, documentation. You need to do qualitative and quantitative arguments to make sure your models, which are mental constructs, are reflecting enough the reality. And we have a template for documentation. So the PCS, or for veridical data science, so I have two terminologies. PCS is these principles my group have proposed. I see veridical data science as a bigger umbrella. It's truthful data science. And we have a particular way following the PCS to do it, but hopefully we'll bring other people in, have their own way of doing VDS, which is a lot broader. So right now, they're kind of the same, but the intention is that they can be different. And PCS might involve too, right? It's very much evolving. We might add more principles. Say for safe AI, we might have something even more urgent for that. And we have been doing recommendation for external causality validation, evaluating clinical decision rules, as I mentioned, many algorithms. We add stability and get better interval result, even improvement on accuracy, and other people have worked on extensions to other kind of subfield of data science.
Can I ask, so
yes.
If the students in your class have been using PCS,
Yes.
then they still would get very different answers, but you'd have like a very good understanding of why.
Well, usually we try to help them. The principle tell you, right, you screen out certain molecules and you aggregate, and you write a document about it. Okay. So yeah, exactly right. For the last 15 years, I've been teaching 215A in Berkeley. And this framework, together with my research, was evolved through teachings. And I'll mention we have a textbook published by MIT Press last year. So now I'm hoping other people use it. There's a free version of the book that you can use. It's actually a very much evolution. In the beginning, I do a little bit, and I switch back to the traditional way. So now it's more streamlined, more coherent. It's helped you to make judgment calls, I'll say. It's not replacing humans, but give you this principle to follow so that you're more principled and actually more faster than kind of mess around and say, what do I do here, what do I there? And definitely a lot more efficient even for me, right? Every project I think about reality check and stability. You have to do a lot more. But it's always ground me in the right direction. Sometimes you forget, right? For this predictability, the first project I did with my former student, Ching-Wei Lim, we didn't want predictability. We went for stability. It seemed just very normal to make the zoo stable for model selection. And took us a month. Nothing was stable. For us to realize we need to use cross-validation, at least get a good predictive model before we seek stability. So now it's very obvious. At the time wasn't. I needed predictability. I thought I'd just go for stability. And in hindsight, of course, constants are very stable. Nobody wants it, right? But at the time, it wasn't clear to me. And we're going for stability only. And then nothing was stable, actually. So you need reality check. And in randomness, you seek stability, you end up with randomness anyways. So this is just a list of things we have done in the last 10 years from like medicine, cancer research, genomics, and computational neuroscience. These principles have always landed us in place. You have to do a lot more. This is not a replacement of many things, but the principles really help us to go to a good place. So let me give you the first case study. Any questions? So this is very much relatively easy application of TCS, unlike the next one I'll tell you took 5.5 years. So I got into cancer research through Hutchinson Cancer Center. They heard a talk by me in 2019, and they thought they have a lot of irreproducibility problems. They validate other people's discovery for cancer. 2019, the replication rate by the validation center was less than 10%. So with very low replication rate, they thought this would be useful. And I already worked with MD Anderson Cancer Center, had a pancreatic cancer detection, well published, the editor liked it, wrote a commentary. But at the time, they just followed. That group completely bought into PCS. So we're writing more proposals about cancer research, breast cancer. So we're there, we just follow PCS. And then I was invited to give a keynote with early cancer detection at DC. I wanted to go in with 2 case studies. So this is a case that Chinaya, who's an oncologist at University of Michigan, already had a paper on ITAC. So went in as a stress testing the pipeline. Redo the analysis following PCS. So they were very generous, so let's do that. Their paper, we're not part of this paper, published in JAMA, it's a top medicine journal. And my people, Tiffany and I work with them. So here's the real world. We have a lot of people. You might need prostate cancer when you get older. You usually do a prostate cancer screening. And this is a new possible biomarker using gene expression and clinical variables. And we're looking at more serious prostate cancer. And in medicine, you always do transfer learning in the sense you always have external validation, a different data set from different population. Because the method is supposed to work not just for your hospital, not for the current population. They use logistic elastic net, right? You have logistic loss function, you have L1, L2 penalties, and they got 18 genes. So this KLK3 gene is the gene that produces a protein that is used by this PSA. So the current standard is PSA test, but the AUC is pretty low. It's only 60%. Right, so you have a lot of false positives. And biopsy is very painful and expensive. So we want to reduce the false positives. Their methods increase the AUC by 21%. So that's huge. This is non-trivial, right? And we went in and talked to Yu Ping, who was a data analyst, said she did some data cleaning, right? thresholding, remove some samples, and we talked to her, said, could you have done differently? She said, yeah, of course I could have done differently. So we did 3 other ways she said I could have done differently, and we ran through the pipeline. And the result only changed by 1 to 2% AUC. So 21, there's no problem, they have huge improvement. And then we went for the modeling stage. They have only 1 model. We added 8 other models like lasso, random forest, and so on and so forth. So we added other 8 models because there's no like physics saying that the gene should be related to the prostate status through linear. We don't have that science. So it's just all machine learning methods data model. So into the 8, and we pre-checked it, had the 6 good methods, and then we interpreted different methods, have different way of interpretation, and we rank them. Right, random forest is very different. It's not really a linear kind of perturbation analysis, but there's an importance measure. And we end up being able to reduce the 18 genes to 8 genes with very similar performance. And for the external validation set, we lose about 1 to 2%, but remember, data cleaning can also cause 1 to 2% difference. So everybody agreed that this is just as good as the original method.
Could you say like there's a 17 to 8 or whatever?
Yeah.
What is the criteria by which some genes were rejected? They're just unstable across the different model selection, or?
Well, so the first step is that we look at the prediction performance on the validation set, and the ones who don't perform very well or the ones who don't have interpretation got deleted.
Yeah.
So we end up with 6.
Yeah.
And each of these methods has an importance measure.
Yeah.
Many linear, but many use MDI, which is the important measure in random forest. And then we just rank all the genes. So you have 6 rankings, and we average the rankings. And then we use that the first, the second, and until we hit 7, because we always keep that gene, then you see similar performance. And then we went to external validation by Hutchinson, so we didn't touch their data. We sent everything over, somebody else ran our code on their data. And that's the curve we see. So as a result, the patent actually just got filed yesterday. So we're now a patent with Michigan, and Shenzhen actually has a startup on the side. So if he brings to the market, I mean the easiest money I'll make. Not dreaming too much, but why not? Yes.
Earlier you mentioned there were also some clinical variables that were featured in the model, but then they're not mentioned anymore.
The clinical variable always used.
Okay.
Yeah, but we're only selecting the genes. Yeah, that's a good question. The clinical variables, there's this prostate volume that we sometimes use, we sometimes don't because it's not available. So all the curves, one of them use it, with it, one is without it. But all the other variables are used. We didn't select the clinical variable. Thanks for the question. Yeah. So what's the value added? Is that we reduce the cost by 55%. Each gene costs about $20, $30. So the group was very happy because they think FDA is more likely to prove an 8-gene panel than an 18-gene panel. And I talked to Jin Yang from Australia. She said her lawyers in University of Sydney wants 5. So in Australia, apparently 8 might be too many. So it varies. Here, they seem to think it's fine. And in theory, we can also do experiments. We didn't to see. I believe that these 8 genes are more likely to be causal. The reason is because it's the next project I'll tell you. Okay, so this is also used for the pancreatic cancer. So stability is really like a holistic sense of uncertainty quantification, right? We don't give probability measures. So later I'll tell you that we actually do some real uncertainty quantification, but it's, if I get to it, but I just need to give a plug to that. We do have formal ways to do uncertainty quantification. I think I might run out of time, so let me just put in a plug for that. So what's behind the next case study, which is much, much harder, took 5.5 years to find genetic drivers of heart disease. It started with Drosophila. Drosophila is much easier to work with. You breed them in the lab, they have very short, relatively short lifetime, and this is very clean genetic signal. So that's why we developed this iterative forest. So this paper came out 2018 with my wonderful collaborators. And so in genetics, the interactions, yes, early on Jesse talked about it's more like correlations, right, related second order. But in genetics, you often have fourth order interactions. Drosophila is a very famous organism. 5 or 6 Nobel Prizes have won based on Drosophila research. And this is called segmentation. It's like, well, similar to humans, when we develop the spine, it's known that in Drosophila, 4 genes have to work together to make the segmentation. So second order is not enough. And the linear made like the statistical way, like you go for the marginal first and call it like second order, third order. It's just computation expensive and also doesn't match the modern biology. There's something called the French flag model in biology. It really says that because when gene interacts through proteins, the proteins have to have enough abundance before they interact. So it's not linear. And decision trees actually capture that thresholding behavior. So people have been using random forests. Yeah, everybody knows random forest, right? Yes. So people already wanted to do, if 2 genes fell on the same path in a tree in a random forest, they probably interact. That's a deeper phase, from mathematical interaction to biological interaction. But the feature is very unstable. They couldn't integrate. So that's why stability is a prerequisite for integrability to humans. So that's why we came in. We added stability. So this is a motivating scientific project for the whole PCS framework. I was doing a neuroscience and this project, try out the idea to see whether it works. So we did soft dimensionality reduction using important index for random forest. When you split, you uniform sample. So we did a weighted sample. We concentrated on the genes that are important from the first run of random forest. Everything else just stayed the same. And we just keep doing it. And then we have to collect all the intersections through something called random intersection trees. The output is interpretable gene-gene interactions, or genes. So it's kind of non-linear model selection. And the computation costs are very similar to random forest. So this is adding stability. So let's see how it works. So this is give you a run of, you start with equal weights, and then you upweight and downweight the soft dimensionality reduction. And then you do this random intersection trees, you collect the paths because there are many, many passes. You map into 0, 1 vector, and you do the collection. And we have an after loop of bootstrap to get stability scores for each interaction. And that's what you see. So here you see that the P in PCS is satisfied. All the iterations, actually, the ROC curve doesn't change. On the right, you have stability score, we cut at 0.5 just to be sensible. And then on the vertical axis are the names of genes or transcription factors that it corresponds to. The red ones are pairwise, blue ones are 3-way interaction, we become recommendation system. And then we submitted the paper to PNS, and the ref said, why do you like it? I said, well, our collaborator liked it. And then
Explain on the recommendation system comment. What do you mean by recommendation?
Means that we can tell people actually have this is the results public. It's for enhancer prediction. We're saying that we identify this 3-way interaction. Can experimentalists do experiment to verify that? So the pairwise interactions, luckily people already did old-fashioned experiments. 80% were already experimentally validated using old technology. And 4 of them, we don't know. We think 2 might be right, 2 might be wrong. We don't know. Okay. So now we're armed with this iterative forest. We wrote a proposal with a big group of people for Trans-Global BioHub Intercampus Award 2018. And I worked with mostly these 2 people, the 2 cardiologists. And then Qing Wang is a postdoc still in Yuan's group, the guy with the red tie. And he went to a startup, the guy with white coat. and Timothy Times, my group, who now University Notre Dame assistant professor. So this is 5.5 years, and many people engaged. So we want to move from Drosophila biology to human biology. Human biology is a lot more complex, right? And so we first phenotype of people's red hair. Red hair is very relatively pure, so it worked. And that's the first phase. And now we went for the heart disease. The heart disease we picked in the proposal, we couldn't find any signal. So I had to switch. We found another one, no signal. We used the UK biobank data. So we eventually end up with this HCM. It's called hypertrophic cardiomyopathy. 5 in 500 people in the US. So it's not exactly rare, but it's not very common. So if you have HCM, your left ventricle heart wall gets thickened. And it's hard for you to pump. You have a smaller cavity. You can faint, you can die. And people believe there's an important genetic component. They think the cell size is the reason why the cell wall gets fattened or thickened. So this is very, very important. I worked with the Drosophila lab, did 40 experiments, but we're not clear about what the phenotype we're going to look at. So we don't have anything to report, even though we think we found an important gene, because we're not clear enough what phenotype we're going to look at. So this is where we're a lot more careful. We know what we can look at, and they have a pipeline to measure the cell size. So here's really try to go causal, right? So we work as a recommendation system to tell the lab what are the genes, so gene-gene interaction should target, and they do gene silence experiment. This is part of the proposal. So we heard a lot of physics here. It's very reproducible, stable, replicable. And you look at statistical causality. I usually follow the Neyman-Rubin framework. And even if you have randomized clinical trial, you still have to make assumption like suitable. There's a unique dose level, and there's no interference. Right? There's no causal influence without assumption. And what we have here is predictability. It follows from causality. And stability also follows from causality. There's something called the invariance principle. Right? But here I'm using the same data. I'm kind of cooking up pseudo-environment by changing the different machine learning models. And they deal with dependencies differently. Therefore, if something searched as important, it's more likely to be important than just use one method. So this is basically what's going on here. And we recommend 5 experiments, and they did experiments. So compared with usual traditional statistical causal inference based on observational data, the model checking is not always done in observational study causal inference. We do prediction screening.
What is model checking?
It's like, does your model have any signal? Right. If you do binary classification, you cannot be the random toss. Then you haven't captured any signal. Right? That's a low bar. But people don't always say, does the model really capture more than just random model you throw at it? And causality evidence in the traditional framework is based on domain knowledge. People just argue I should have this continuum independence and this and that. We do stability. And we never claim causality, we claim as recommendation and for follow-up evidence. So a little more conservative, and usually the causal observational study just say I found causality. And I find it very hard to know whether the assumptions, if I read papers, they're reasonable or not. Usually there's not a lot of discussions. I usually look at the person, oh, this person is a good scholar, I trust it. Basically, I cannot just look at the paper and say, oh, this is well-supported assumption. So it becomes a much bigger kind of assessment on scholarship than just that's paper. They don't write a lot, and I don't know the domain knowledge. They don't have the documentation. So I find that a lot of causality claims, I think the assumptions are shaky. Observational causal study is really about assuming causality and estimate the size. But the conclusion doesn't always say that clearly. It becomes proving both the causality and estimate the size. So what we do is we want to aim at high yield experiments. We don't claim every experiment will be successful. And we Why we believe this is useful and have high yield is because we significantly expand uncertainty account. So if you pass that, it's a higher bar. Therefore, it's more likely to be real. Yes?
So what do you mean by high yield?
It's like if you do 5 experiments, I don't want only 1 or 0 to find causal variance. That means the number of experiment versus the discovery. The discovery versus number of experiments should be high.
Okay.
Yeah. Because people, not all experiments successful, right? That's a known fact. And they usually just think about it and do something. Here we give them some AI or data science support. They will still think about it, say we do look at databases and we talk to them, but we really reduce the scope of design space. So this is the data UK Biobank data. What the data 0, 1, 2 means, and most of us share the same, we have 2 double-strand DNA, and most of the base pair bases are the same for all of us. And if we agree with the majority, you get 0. Each column is a location. If you have 2 alleles different from the majority, you get 2. If you have 1 allele different, you get 1. This is called the dose matrix. It's how different you are from the population. And a lot of time people believe these are the reasons people have disease, because you're different. Most people are pretty well. So, and the HCM labels didn't work. So we had to go to a new label, which is going for the volume, the hard form. And we have to normalization, and we did dimensionality reduction. So let me give a quick run. We use unrelated individuals, therefore they're not family related. So we just did a random split. And then we use GWAS. Oh, important. We need to satisfy the P, right? So if you do regression for the size, we don't know whether it's noise level. So we end up binarize the size into the top 20 and bottom 20. Also, we made a clear signal, and we did stability analysis. So if we couldn't beat the 50-50, then we have no signal. We shouldn't interpret, we shouldn't recommend. And we beat it by 5%. This is a lower supervised learning problem I ever worked on. But it's consistent. If you do 15%, 25%, and you do different methods, they're all consistent. So this gives me a sense that this is probably real. This is a very hard, complex problem, right? People hard-sided, exercise, eat differently. This is people in their 50s, 60s, not babies. If babies, maybe the signal will be clear, the genetic more at play, but here, there are a lot of environmental factors we cannot control. So you shouldn't expect very high predictability, that's what I'm saying. But you have to beat 50%, considering which we did. And then we did also dimensionality reduction using GWAS marginal, and we used 2 softwares and we did a union. And we have to add more stability to RF because it's very signal, I won't go to details. And then we prioritize interactions. And then the biological experiment. So as I said, so we need to have an integration of computational and wet lab because statistics is not enough to really make conclusive decisions. And we need domain knowledge. We look at the annotated databases. So because the failure with the Drosophila experiment, now I interact with doctors or experts. I give them 3 lists. One is our preferred one middle ranked one random one for them to choose, instead of giving the ones we picked, because then they will have confirmation bias. So that's better signal of confirmation than just, oh, they always usually can make up some story, right? So we try to fool them a bit with negative control. So far my collaborators haven't failed. They always pick the one we liked, our model picked. And we did a lot of extra data analysis. So here's the experiments. These people died in Stanford. This is stem cell. They're round. They're not like elongated as the real cardiomyocytes, the muscles. And this is this like this machine built by Chen Ru. And you push all you do gene silencing, and you push all the cells, and you automatically count the cell size. You basically find segmentation. We checked with random check all of that, and we find some problems, and we fixed it. So in the end, just give you a quick story. Oops. We did 5 such experiments. We also, by luck, we got 80% success. We found 2 gene-gene interactions. which is very, very hard. This is the frontier of functional genomics to find gene-gene interactions. And with experimental validation. We compare with conventional methods, convince the referees that we are better, because this is new methodology, and then finally appeared about 2 months ago in Nature Category Research. So this is my main collaborators. I'm hoping that one day there will be a drug targeting one of those gene-gene interactions. And the response to the first submission was 38 pages. So it's a really long process. Big shout out to Chengru and Tiffany, just never wavered to finish this project. It was 5.5 years. We started right before the pandemic. So to summarize that, the code, I'm hoping that other people looking at other diseases can use our code, right? Because this UK Biobank, every user data, we care about this particular heart disease. I'm hoping other people use it, but I need to find other collaborators try it out before people adapt it. And we did a lot of other data analysis too, which I won't talk about, gene enrichment.
In those examples, could you comment on what stability analysis looks like? Like, what do you mean?
So we first did, we didn't have data cleaning because UK Biobank data is already cleaned. We don't have the raw data.
Yeah.
So what we did is that we did dimensionality reduction. We used 2 marginal called GWAS packages, both based on linear style. One is called Plink, one is called Manbold. We took a top 1,000 gene, and we took a union. So we didn't want the first step to be decided by a particular software, because they are 50% different.
Yeah.
So we took a union because we don't want to be taking the intersection at that stage. And then we did the iterative random forest. It's already a lot of stability baked in, but at the SNP level, for the red hair, we did at the gene level. But for this, we did at SNP level. Actually, the SNP level, nothing is stable. So we collected the interaction at the gene level. So that's added stability. And then we did the predictive analysis. We did the binarization, 20%, 15%, 25%. We couldn't do infinite possibilities, right? And then for the final test result, we actually did a robust statistics. We did train mean because we delete the very big cells and very small cells, because some big cells are actually clumps. That's our discovery. The cells don't always separate. So we look at a lot of videos. They can be cells stuck together. If you count it, they're really going to mess up your measurement. And there's also small cell sizes might be fragmented because the force is too strong, break the cells. So we very much did robust stability in comparing this average cell size. We see a whole shift of cell size when you knock down some genes. So there's other different steps of stability. We didn't do formal intervals, anything like that. I find it very, one friend said that he likes the way it's more holistic than very, because it gives you a lot of freedom to do different things. So I think both are useful.
I notice in your examples there's like some part of stability analysis that's kind of statistical, and another part is you're paying a lot of attention and you notice things that are wrong in your data. Like you see those as like...
Yeah, so we watched a lot of these videos, and then we saw this humongous cell which got counted like 38 times, and they didn't know. And then that was our big discovery. They were not too happy when we said, I want to work on an experiment, because they have done it for a long time, and my young colleagues said, you will have, and so anyways. But they were very nice, and they let us do it, and they were very happy with it. So we designed the experiment. And they added more forces to break these big clumps of cells. And we did also train mean, so we don't get it back. So that was like old-fashioned statistical detective story. You'd be there. Right before the pandemic, I took my people visit Stanford for a week just to see the lab, meet the people. And then the pandemic happened, everything shut down. So that was February of 2020. So we did a bit theory, because after this we really think RF. We made a theoretical version of RF, because too much random to analyze, so we changed random forest by making stability in the way that we only count the split if the drop of impurity index is larger than a fixed amount. So that we can analyze mathematically. So this paper came out, I think 2020, probably 2022. Then we can show that this is, I have a hypothesis I will call discrete before continuous DBC, I shall give a name, is that in statistical work, also in causal inference, I believe that you should go for the discrete structure first. And if you do it together, you mess it up. So this is the first mathematical proof we're able to do. So we have, I put it there but it disappeared. So you have a regression function with a linear combination of Boolean functions. And we can show this theoretical version RF confine the interactions without ever explicitly estimating the threshold, which is continuous, and the linear coefficient, which is continuous. So this proves that the discrete structures actually are easier to estimate, and you should go them first before you go to continuous. When you do Gaussian graphical model and the Perl framework, you actually mix them up. I think it's not a good idea. You should really go for the causality. So that conjecture came up. I went to BioHub because to get the money, I have to go 50% accept and use accept. So there are a lot of talks to biologists now using computation biology. And they figured out a lot of things about biology. So I said, well, these people doesn't always need computational models. It's just a good logical thinking. So I said, and there's a paper from 1964 by a guy called John Palade called Strong Inference, who made exactly the same point, that continuous things are flimsy and discrete things are logical thinking are more stable. So this is the first proof we had that in a rigorous condition, we showed that you can go for the interactions without ever estimating the continuous parameters. So I think there's something a lot more to that. Even you look at max micro, you have uniform from 0 to theta. You can estimate theta at 1 over n. And for continuous Gaussian, it's 1 over square root of n. So there's something there already. When you have discrete signal, it's much stronger. And you mix it together, and then we are worse off. So this is, I hope somebody will follow up on that. The conditions, we made it pretty stylized. We assume all the features are independent. Of course, in genetic they're not, right? But that's the best we can do for the first cut. And then the interaction terms, we assume they don't overlap. Of course, in genetic they overlap. So we make the simplest assumptions. But still, it was really neat work by Merle, Shell, and Hill.
5 minutes.
Yeah.
5 minutes left.
5 minutes, okay. So uncertainty quantification, I'll say that we have this book, and there's a free version on PCS by MT Press, and Rebecca is really brave to work with me when she was first year PhD student, and now she just left Utah to go to a company. And we have a follow-up paper, just take home. We reduced the best conformal we compared by the weights by 20%. We're more adaptive in terms of subgroup coverage, conformal inference, right? And actually it's a new form of conformal. We have subgroup result. We can speed it up, do auto better than conformal for deep learning models because we do bootstrap and it's too expensive. So we have speed up, and we can show that a slide multiplied version is a new kind of conformal, and the exchangeability, we have the coverage, but we have other things to do. And I see this as a certification of whole data science cycle. It's not 100%, but it's a certification. And we have that neuroscience, protein prediction, improve UMAP. I talked to Dan about it. Hope you try our way because I think TSNE and UMAP are too flexible. We have these artificial clusters coming out. And we can already improve that uncertainty coefficient by combining with quantile reduction. So on top of 20%, we have further 15%. But we need 2 calibration parameters. And resources, we have some software to help you parallelize the stability analysis. Oops. Wow. I think I should finish. It's not stoppable. Yeah, but the free book, let me just say it again, vdsbook.com. I had a QR code, but it's not allowed.
Doesn't let you go back?
No. Comments and critiques, I guess. You seem to know what I should say next. Yeah, it's out of control. But maybe I should, because I do want to show you some theoretical problems. Yeah, open problem. Let me go there. So first, can you remove the condition independence? And can we synthesize different notions of stability? I think there has to be connection. Which notions are equivalent under what conditions? And that would be really great. And I feel like really PCS really this plural philosophy, right? Most of the time we just get optimality. I never see optimality in real problems. It's always do the best you can. So can we bring this plural philosophy into theorems to show that it's just more resilient, it's more robust, right? You have to have misspecified model. You won't see it if you only have one model. So you need multiple models and consider some methods, right? I like spectral clustering because people show under manifold, it adapts. When you have stochastic block model, adapts, right? So those are the result I'm looking for is an empirically successful method can adapt to the data structure. And that's a form of theoretical stability. And the last one,
Sorry, can
I
you
think,
just say that last sentence again?
called theoretical stability.
Yeah, an empirically successful what?
Empirical successful is the plural philosophy, try multiple things. It's really like this common sense that with PCS, it's good data science hygiene and using this common sense that when you have a serious illness, you always ask for the second opinion. But you go to a good hospital, a good doctor. So that's what we do. We screen to find the good doctors, the good models, and we aggregate. That's just very, in hindsight, very common sense. 10 years ago, it was not common sense to me. I struggled and I didn't see and make it coherent. But in hindsight, I think who doesn't want reality check if you work with data? Who doesn't want to remove some arbitrary, reasonable human judgment calls from the conclusion? Like 20 of us here, we have 20 different conclusions. It's hard to defend as we pursue truth as a scientist. So I think before we didn't know, I didn't know, but now I think that there's so much recognition now, and this takes longer to implement, but I think AI will actually help, can automate a lot of this process, which would be harder to do if you just manually. So I think this is really AI. So the book is, some people in China already made a VDS agent. I haven't checked it, right, to make it automated. Right, so this automatic data science pipeline are coming up at different places. And I would like to see people actually do automatic this check-ins, this stability. And journals can also, I was at Flight, I already have a cryo-EM competition. They were very open to say that maybe people submit their results can just fill out a Google form and say in your modeling where you did reality check, where you did stability and make the referee a lot easier to read. You can write the paper the same way you used to, but just collect, like your question, where did you do stability? Right? It's really embedded, but you can just fill out a Google form and put it together. And this should be minimum for every data science project. But it's still, I think with the book out, I was very encouraged that we have run 3 workshops, which that's what I couldn't show you. Yeah. So we ran 3 workshops. This is 2 or 3. Well, there are only 2. Yeah. There's one also run by students. So we're probably going to run one in Paris next year, and then I'll probably use this network and to let people know. And we're going to do a webinar, bi-weekly webinar. We really design in biology. So this is an area, I think, biology that this probably will benefit the most. In physics, people probably a lot more rigorous and the data, hopefully. So I think the low-hanging fruit is really biomedicine, where these ideas, I think, will really play a positive role. And just to show off that we do have that's the book, right? And we had a very positive book review by Harvard Data Science. So just, and we did a session at our annual meeting. It was last day, 8 in the morning, and 20 people show up. So it was really nice. That we probably going to. So one person actually they find a little hard to integrate, so they just did the offshoot project. They did a cargo type of competition, didn't change the course material, but did a competition with the students following this framework. So that's the easiest level. Oh, just send a link to your students. It's free. You don't even have to talk about it, right? So that'll be really helpful. And then, you can take some of the materials. One chapter is on data preparation. That's what here people find really useful because there's no other book talk about it, about GitHub and all of that setting up. And you can choose and pick all the codes and the data set on the archive. We have 5 kinds of exercise: true and false, conceptual, math, data analysis, and coding. So all of that is all free. And big thanks to MIT Press to allow us to do that. I hope they're making some money. They don't let us to share PDF, but the web version is fine. We're talking to people in Tennessee. They work on K-12. Maybe we'll have a high school version of it. Because it's very accessible. Not a lot of math. There are a lot of great math books. So this is try to fill in the gap. Thank you.